This method can help elucidate the molecular mechanisms underlying the regulation of candidate target genes via epigenetic modifications during fungal pathogenesis in plant pathology. Chromatin immunoprecipitation sequencing technology can efficiently detect DNA segments that interact with histones and transcription factors in the whole genome. Compared with other methods, it can improve efficiency and resolution. Begin by adding 100 microliters of liquid complete medium to the oatmeal tomato agar plates using a 100 microliter pipette. Using an inoculation loop, scrape the mycelia of the wild-type strain and the knockout strain. Collect the mycelia debris and transfer them to 250 milliliters of liquid complete medium. Wash the fungal hyphae with 500 milliliters of 0.7 molar sodium chloride solution, then collect the fungal mycelium and weigh it. Add approximately 1 milliliter of lysis enzyme permeation solution per 1 gram of the fungal mycelium. Place the hyphae for lysing at 28 degrees Celsius for 3 to 4 hours with shaking at 150 rpm. Wash the lysed hyphae with 50 milliliters of 0.7 molar sodium chloride solution. Centrifuge the sample and discard the supernatant carefully. Resuspend the protoplasts in 20 milliliters of 0.7 molar sodium chloride solution. Add 55 microliters of 37% formaldehyde drop by drop to 2 milliliters of sodium chloride buffer.